Hello, and welcome to another Wolfram Tech Talk. I'm Arben Calzici, a content developer and instructor with the Wolfram U team, and today I'm talking to Daniel Lichtblau, a senior symbolic algorithms developer. And he'll be telling us a bit about the possible emergence of a new COVID variant.
Thank you. Thank you, Arben. So, a couple of words. This is joint work with my colleague John Castle, who knows a lot more about biology and genomics than I do. So people have questions. I don't know what to say. Send him email. I could try to answer, but so this actually, I was asked if I could do one of these interviews about some research project that might be of interest, and this is one that John and I had recently written about for the Wolfram Community Forum, and if I can get the mousing gestures correct, this is what the beginning of the Wolfram Community post looks like. And in this post, I allude to some incidental/accidental things that happened during the course of working on this project, and I'll say a little more about that as we go. But the idea that I had originally was, since Australia is an island country and has restricted the inflow and outflow of people during the pandemic, I thought it might be nice to get some sense of how COVID might be changing in that country. This was just at the time Delta was starting to hit. There is some migration into the country, of course, and that seems to be what brought in Delta. So what I had is a snapshot of what took place just before Delta. And so I'm going to focus on that a bit. Let's see if I can get the mousing. Okay, and so what I wanted to do was compare what we see in Australia with the main variants of concern. So I got all these sequences from, I think it's pronounced GISAID, might be GISAID, I'm never sure. And I took recent sequences from all over the world for the main variants. And the stipulation was that I restrict the count to be in the 400 to 500 range. I think one was a bit more because a bunch had come in over a period of one day, and I would have had to take either 300 or 600. And so I did that, and I load all these sequences, and you're going to see I used alpha, beta, gamma, delta, the 4 variants of concern, and also Kappa, which turns out to play a key role in this melodrama, really more of a farce. But I didn't originally do that. Originally, I used other variants. Some were variants of interest. Some are no longer on that list. I used what was called the New York variant, might be iota, I can't remember. I used the 2 from California, and I think I also used mu. And from what I could tell, none of them were in any way related to the what I saw, and I'll explain a little more of this as we go, but what I saw in Australia, so I removed those. After doing a lot of this work with John and getting some really neat results, seemingly, I did some background reading and learned that the Kappa variant was not the same, or even it was close to, but not that close to Delta, and that it had appeared in Australia, and that's when I realized that not using the Kappa variant in my original analysis meant we were getting results that were much stronger than they really should be. So that was a bit of a mistake, and after shutting myself into a closet and sobbing for several minutes, I went back to work using the Kappa variant, and that's what we're going to see here. So
I read in the blog post that you're using, and I think that's what we're seeing here perhaps, you're using alignment-free sequencing. So usually, to my limited understanding as somebody with a physics background, when you compare genomes between different bacteria or viruses or whatever organisms, you have your little sequences of DNA, and then you line them up and you look for differences. So how does this sequence-free alignment differ from this kind of more traditional method of alignment?
Well,
Or alignment-free
yes.
sequencing?
in two ways. The most important, I think, for our purposes, is that I understand the alignment-free methods, whereas sequence alignment is not a game I know how to play. Fortunately, John Castle does. Another difference is that the alignment-free methods tend to be faster. You can throw more data at them. You get cruder results, although as we're going to see, they're not that crude. And they give you some ballpark ideas of what you're dealing with. You can then go to alignment-based methods at a smaller scale. The alignment methods can be slow, and you can't just compare thousands upon thousands of sequences, each to every other, but you can compare pairs, and you can compare many pairs, and get some finer sense of what you're looking at once you narrow the field down. So what I did was the narrowing.
Okay.
And I'm going to show, well, we have on the screen one such alignment-free method called multidimensional scaling. What I do is take the genome sequences, convert to something called the frequency chaos game, FCGR. I forgot the R.
Regime.
Man, that's bad. Something like that. And so this creates a square image of pixels that can be seen as a signal. And from that, we can use image and signal processing methods to cut back the dimension, because this multidimensional scaling MDS, it's also a dimension reduction scheme, but it works well if the input has dimension in the, say, low hundreds. If it's in the thousands, you're dealing with hardcore linear algebra that will bog down.
Right.
So, and it's based on, if you're familiar with it, something called the singular values decomposition. Very few people are familiar with that. It won't be on the quiz. And so what we do is project down to 3 dimensions, doing all this signal processing manipulation, and then we plot the points. And what we can see is that the pink kappa and the blue delta are kind of close to one another. I can rotate. And the others are pretty well separated. We then use a similar set of signal processing tools to build a phylogenetic tree. There are other ways of building phylogenetic trees, and I won't say that this one is the best possible, but in practice it works well enough. And in particular, and this was to my surprise when I first learned this maybe a year ago, it is good enough, as is this picture, to actually distinguish variants of COVID from one another reasonably well. Again, there's a fuzzy boundary between kappa and delta. And there's a reason for that. They share a number of mutations. So let me see if I can, so here's the phylogenetic tree. Well, it's just a tree that shows, among other things, the reds hang out with the reds, and then there's this big gap. It doesn't look like a big gap, but when you see that it's a subtree that is apart from the red subtree, where we see the oranges, which I think it was either beta or gamma. So then we get to the delta and the kappa, and I forget what, I think this is beta we're looking at. And you can see that they're really reasonably well separated subtrees. It's not perfect, and in particular, it looks like the last of the betas, I guess, or gammas is hanging next to the first of the other.
Yeah, but that really is startlingly good, given the method you described for getting these differences listed. That's very surprising.
Yeah, I was happy to learn this. There are perhaps some confounding issues having to do with how the sequencing is done. Apparently, from what John Castle told me, some labs will lop off a beginning or ending sequence. And that was something we actually had to deal with when we did the alignment-based methods to get the aligning to work.
Is that because, like, I think in genomes there's usually some kind of signal that a sequence is starting and then a signal that it's ending, and are they chopping those off?
I have a 2-word answer for that. Ask John.
I will.
I think that's it, but I don't know for sure.
Okay.
John was the expert in all things alignment-based.
Yeah, but the point is it might have generated some difference between the sequences.
Definitely.
Right.
Yeah.
Okay.
And it might even be lab-specific as to the protocols that they use. So then we throw in the Australian sequences, and we do some of these similar things. Now, for the Australian sequences, I had to come up with a coloring scheme, and I think I had gathered these, I believe these were all during, well, let's see. Yeah, they go from January 1st into, well into July. And there's about a 2-week lag. I was doing this late July, but yeah, the time of collection, there's always a lag between collection and the actual sequencing and then uploading to GISAID. So we only got into early July. So I tried to color the Australian sequences with shades based on date, and in a way that wouldn't interfere too much with the 5 colors already being used. And what I thought I saw was some greenery up here from relatively recent Australia, and another set over here. And here is where the alignment-free methods did not do as well as I would have liked. And there might also be some over here. It turns out, I thought originally that these were the sequences that were going to delve into further later, and I thought that they were their own new variant. That was before we had put the Kappa into the mix. What I later learned is that they are pretty close to Kappa. They're in here. And they're pretty close to Kappa because they actually are Kappa, but they're not entirely perfectly Kappa. So what we saw in Australia seemed to indicate an evolution of a Kappa strain, possibly into a dead end, because once Delta hit, it sort of spread and probably by now has taken over.
Yeah, it is dominant, yes.
But Kappa hit in Melbourne, and they had a bit of a spike, but then that I think died off. Delta came in mostly through Sydney and through the capital region, which I believe is surrounded by. So Sydney's in New South Wales. The autonomous capital region with Canberra is, I believe, surrounded by New South Wales. Excuse me, and that's where Delta first hit. But I would imagine it spread throughout, just as it spread elsewhere. So anyway, yeah, so we can go up, and so what we're seeing here, in the phylogenetic tree, I use coloring for the dates, but I also use the names, short names for these, just to get to know where they're from. So Vic is Victoria, which is the province with Melbourne, and those are inside the Kappas. And now here in order to get this to complete in reasonable time, I culled out, I decimated, basically. I think I took 1 out of every 4 from each of the groups.
Right.
So we're not seeing quite so many sequences as before. And a lot of these older ones are just, they're mixed in with, I think it's, purple. Whatever I said about it before, I think that's alpha. Well, there should be a legend at the top.
So these are the data.
Alpha.
These are the data from before. And then you've taken about a quarter of the data from Australia and put these in here for comparison's sake, so we can see where they lie.
Actually, I think I also took a quarter of the others.
Okay. Yeah.
Skip. Yes, I think I did that with all of them. So I've tried it going as much as a half and then doing the other half, and the results always look about the same. So
Right.
it's not.
It's a representative sample. At least.
And all right, so we see there's not much in with the reds, and there's a whole lot here with Delta. And although these branches look pretty long, they actually, when we do some sequence alignment, they actually really are Delta. This is where the alignment-free method gets a little bit weak. It's probably also where you need to know about the vagaries of pruning the initial and final parts, or not pruning them from the sequence, because that in effect, if you leave those in, it adds noise. And that can make these appear artificially further away from
Right.
the main body of what they are, in this case delta.
Right.
So you get these sort of longish looking tree limbs that makes you think they're farther away than they really are. At least they're mixed in with delta, which is as they should be. So going on down, or going back up, I should say, we'll see the, I believe this is where we're looking at Kappa. Do I have my colors right? I can't tell if we're looking at gamma or Kappa. I believe this is Kappa, and the Victorian sequences all look like they're really mostly close. There are a few at the top, maybe not. But it turns out, again, they're a little bit further than you'd think. We had to use the sequence-based, the alignment-based methods to see that.
Right.
I sometimes wonder whether I'm getting colors well enough. My eyes. Okay, so now we're on to the part of the talk where I channel my inner John Castle, because this is his expertise. And I'll mention as an advertisement for the Wolfram Function Repository, where I'm the lead curator, I don't know why, that John contributed much of the functionality being used here. You can see as resource functions. They are in the Wolfram Function Repository. I should say the same about the functions I used in the alignment-free method. The multidimensional scaling, the sequence to FCGR, the phylogenetic tree building, all of those are functions in the Wolfram Function Repository.
So those are all in there, and then we also have things as specific as remove degenerate sequence differences, alignment to position differences, and align nearly identical sequences. So these are all just built into that.
Yeah, John built the tools here specifically so that people could do this kind of processing. And in ways, this notebook is can serve as a blueprint for how others can do similarly. And it's really neat. I was actually able to use some of his code. There was a time when he wasn't available. I think he had like a half week of wall-to-wall meetings. And I was able to use some of this code and then just run past him the results.
Okay.
Oh, so here we are in the alignment-based code. So let's scroll downward.
Yeah, and it seems like in the beginning you imported that data from GISAID, and once you've done that, you're just totally in the notebook doing all of this, right? There's no need to leave that for any purpose. Okay. And that's thanks in large part to the functions that John contributed to the resource function.
Yes.
Yeah.
The
function
Wolfram Function
repository.
Repository. Yes, the only reason I would have to leave was for a coffee break.
Not in notebooks yet, unfortunately.
Yeah, not in the notebooks yet. And so what his code allows us to do is, among other things, find similar sequences. We compare to the reference sequence, which was the first, I believe, genome sequenced out of Wuhan in, I think it was January of 2020. And the whole world of COVID has mutated away from that since then. But by using that as a baseline, we can find the mutations that are common to, say, Delta or Kappa,
Right.
or any of the others.
Yeah.
Now, we learned from the pictures that, and from some other analysis that's not shown here, that we really weren't looking at recent sequences in Australia that might be related to alpha, beta, or gamma. Some of the older ones, sure, but the recent ones, the relatives were Delta and Kappa. And what we found that turned out to be of interest was not the original grouping that looked like an outlier, but this other grouping that was fairly close to Kappa and really part of Kappa. And we were able to, well, this is all initialization code in a sense. We use it later. But what we were able to do is isolate common mutations mutations that are common to Kappa but not to that cluster, and there's very few, but there are some, and vice versa, mutations in the cluster not common to Kappa. And then we're able to figure out, using the coding sequence regions, and which is data that John can download and did, and also using the alignment code, the code that computes amino acids from nucleotide triples, we're able to figure out what changes in proteins that get coded for.
Right.
And so that's what all this is about, and I'm going to mostly gloss over it because, again, it's more John's area. But we use this to get some specific genomes that were representative of the Victoria cluster, and that's cluster items 1, 2, and 3. We were able to get specific items from specific genomes from delta, kappa, and alpha, and we're able to use the differences. And from these, we're able to come up with a bunch of the, we're able to find all the genomes in the cluster that seem to have common differences, or have most of the common differences common to that cluster not found in the other variants, in particular not found in Kappa.
So using this method you've described, comparing to that reference strain, you could say, if you just looked at delta versus alpha, beta, gamma, you could do something like find out where the differences in the sequence are, convert those to amino acids, and then say, oh, okay, that's coding for the spike protein, and we see there's a difference between these 3 variants and delta in terms of, say, the spike protein.
Yes, very much.
So then you can do that same thing here to see what what different proteins are being coded for these Australian variants.
Yes.
Okay.
Exactly.
Okay.
And I mean, I know essentially nothing about proteins other than when you're dealing with, say, one substitution of an amino acid. I guess you can consider the thing to be, the two to be the same protein essentially, but with a minor difference. That minor difference, however, can have structural and or functional meaning.
Right.
So, it's still the spike protein, but you might have different variants of that behave in slightly different ways. So what we've found is that there are, in this Victorian cluster, there were 11, let me see if I have my numbers right. Sorry. There were as many as 11 common mutations. 18 genomes had all 11, 28 had 10 of the 11, 5 more had 9, and I think that it's that group, so 28 plus 18 plus 51. I think we worked with those 51. We accepted that was our cluster. And there's a pretty fast drop-off after that. After 9, first, there's only none with 8, only 1 with 7, and then it drops off to 4.
Right.
So
it clustered pretty tightly at that high end of the number of mutations.
Yes, exactly.
Okay.
So, and here is a histogram, I guess, bins that show that. And so now we figure out which of these 50-odd genomes comprise our cluster, and we can then key in on differences between those and the representatives from the variants. And again, at this point we only looked at delta and kappa. There were essentially these were unrelated to the other variants. And so the background diffs, I think, has to do with removing all mutations that are mutations from the original reference genome that are basically common to every one of these now.
Yeah.
And so we remove those from consideration. And so we see that there are a bunch of protein coding differences between this cluster and what is nowadays sort of the common core. But many of these are we will see many of these are shared by Kappa. In particular, I think some are shared by Delta, many are shared by Kappa, and so we have to remove those. It's where what we say here, there's a significant overlap with the Kappa variant, but it's not 100% overlap. And so what we do is we show what's in the Australian cluster but not Kappa, and vice versa, what's in Kappa but not the cluster, and we see that there are indeed a few differences in the protein coding.
Yeah.
And so that's where we ended up with this. I think that is the bitter end. And, in some ways, I view this as snatching victory from defeat, because, before we thought we had really uncovered a new variant, and then we realized, no, we have Kappa. But then we realized, well, this isn't actually quite Kappa. It's a little bit new.
Yeah.
And what I surmise is that in isolation, variants themselves can and will mutate a bit. Whether these mutations were beneficial or not, I can't say.
Yeah.
Why some mutations were lost from Kappa and gained, and why some others were gained, I can't say. Maybe the ones weren't really lost. Maybe what came overseas to Australia were uncommon forms of Kappa that already were, that were missing a couple of mutations. I don't know.
Yeah, there are a lot of questions about selection pressure that one
Yeah.
could ask. Yeah.
But, I mean, at the end of the day or year, as it turns out, I think it probably doesn't matter, because Delta has superseded Kappa. I tried, in some later work, I tried to get more sequences, more recent ones from Australia. What I learned is that a lot of sequences coming out of Australia are not coming out of Australia. That is to say, they're staying in Australia. They have their own data repository.
Right.
Not much is getting uploaded to GISAID. Between late July and late August, I don't know if any got uploaded that came from Victoria. So I couldn't really say more. I could have probably tried to get permission from this consortium in Australia to get at their sequences, but this isn't really part of my day job. And I sort of thought I had other things I needed to attend to, like, fixing a few bugs here and there in Wolfram Mathematica.
Yeah. Somewhat important, yeah.
Yes. So that's where this landed, and it was a fun project.
Yeah. I can definitely see what you mean about this kind of serving as a blueprint for doing this sort of analysis in the future. Seems like, yeah, with just a few, well, not a few, but with just some sequences being uploaded and taken into the notebook, it seems like you can do quite a lot in terms of figuring out what the real differences are. Again, in the protein specifically, it's pretty amazing that you can go from just those sequences to that at the end. These are the mutations in the proteins.
I was amazed too. I mean, frankly, I knew John was good at this, but when I had the alignment-free stuff up and running, and again, when Kappa wasn't in the mix, what I had looked like it was pretty stark differences and fairly interesting, and John got excited by that. And then the speed with which he came up with this set of tools for doing the alignment, and the quality of results, it absolutely astounded me. Which is good. I mean,
yeah, it's a good astounding.
Yeah, exactly.
Those are hard to come by.
Agreed.
Yeah. Well, thanks a lot, Danny, for that very interesting talk about analyzing a possible new COVID variant. And I guess variant is kind of an arbitrary term anyway, right?
Yes, thank you very much. The last person who tried this fell asleep, so thank you for staying awake.
Yeah, it was a real struggle. Yeah. Well, thanks a lot. That was very interesting, and I look forward to seeing more work on this subject, if there's more work to be done, in your opinion, if we get more data out of Australia, at least.
Yeah, I can't predict, but sometimes something interesting arises, and
You've got the framework.
Got it, and I know how to get a hold of John.
All right, yeah, that's a useful resource to have. Well, thanks again.
Thank you.
All right. This has been another Wolfram Tech Talk from Danny, who told us about analyzing different COVID variants. And I'd like to note that we have a free Wolfram U course available to you about doing data analysis and visualization of COVID-19. So that includes kind of the spread, some modeling, stuff like that. I think it'd be quite interesting if you found this talk interesting. Stay tuned for future Tech Talks. We have quite a few more coming up. Thanks, and have a good day.